Okay, so now we're going to talk about proteins, which are pretty exciting. So proteins have many structures, and these result in a wide range of functions. So proteins account for more than 50% of the dry mass of most cells, which is actually pretty impressive. And proteins, just because they have the wide range of functions and many structures, they are often what you might refer to as the building blocks of the cell. Protein functions include structural support, storage, transport, cellular communications, movement, and defense against foreign substances. Or defense against the dark arts, if you're into Harry Potter. Just kidding. Alright, so here is a list of protein functions. So here's several different types of proteins. So we have enzymatic, structural storage, transport, hormonal, receptor, contractile, and motor, and defense proteins. Then some functions and examples. That might be a good chart to look over. And then enzymes are a type of protein that acts as a catalyst to speed up chemical reactions. So you guys probably talked about enzymes before, and you talked about catalysts before. Catalysts are an agent to help speed up chemical reactions. Like the reactions would occur on their own, but the catalysts or the enzymes help bring the two reactants together and help stabilize them in certain positions so that the reaction will actually happen quickly instead of almost by chance, which would take a lot longer. So enzymes can perform their functions repeatedly, functioning as workhorses that carry out the processes of life. So here's an example of an enzyme, and with a reaction that we talked about earlier. This on one side we have a dehydration reaction, and then we have a hydrolysis reaction. So here, let's start with the enzyme, and then we have our substrate of sucrose. So two carbohydrate monomers, this enzyme has two active spaces, or spaces where those two monomers will fit right down into exactly right. It'll bring in a molecule of water that will do the hydrolysis reaction, and then I'm sorry, it's just a hydrolysis reaction in here. But then it'll do the hydrolysis reaction and then release the two monomers by themselves. And then most enzymes actually work in reverse, but this one is showing working in one direction, and it is a hydrolysis reaction. Alright, so polypeptides are built from the same set of 20 amino acids, which you guys talked about before, and a protein consists of one or more polypeptides. And so let's talk about amino acids. And these are organic molecules with carboxyl and amino groups. So basically what that means is carboxyl is in the yellow here, the carbon with the double bond to the oxygen in the OH group. So that's a carboxyl group, and then we have the amino group here in the orange. So that's what amino acids generally have. So amino acids differ in their properties due to different side chains called R groups. So here's the R up here, and here are all the different options for R groups. So here we have like glycine, alanine, valine, serine, threonine, aspartic acid, glutamic acid. You guys probably, it might be like sound familiar to you at some level, but all amino acids have these three components: the amino group, carboxyl group, and this R group. And so all these build different proteins. So here are all the different types, and they all have different properties, and that's one reason we kind of talked about earlier. Causes how different proteins are going to fold, how they're going to be different shapes because of how they interact with each other and how they interact with the environment, which is usually aqueous. So that's pretty awesome. But anyways, so amino acids are linked by peptide bonds, which we talked about. And a polypeptide is a polymer of amino acids, so lots of amino acids joined together. Polypeptides range in length from a few to more 1,000 monomers, and each polypeptide has a unique linear sequence of amino acids. So here is a dehydration reaction where you're forming a peptide bond, right here, peptide bond. Yes, so a functional protein consists of one or more polypeptides twisted, folded, and coiled into a unique shape. So here's an example. Basically what that means is you have lots of different, because of the way they group together, they'll form different shapes. We'll talk about this in a second. But when they're all together, you'll make a functional protein, like the whole burrito, like the whole thing together. So for example, here's a picture of a lysozyme, which is a type of enzyme. And you can see all these little ribbon structures here, which are kind of how all the little amino acids are joined together and the shapes they're going to make because the way they're joined together. And then when they're together, they make this functional protein. So here's the ribbon model and the space filling model, kind of what it would more look like. So the sequence of amino acids determines a protein's three-dimensional structure, which we talked about briefly before, and a protein structure determines its function. Surprise, surprise. Okay, so for example, an antibody protein would have a certain structure that fit exactly a protein from a flu virus. So part of an antibody's function is to recognize proteins or other things from, foreign substances or like viruses or bacteria or anything that's not supposed to be in the body. And even some things that are supposed to be in the body, but it recognizes them. And it recognizes them because it has a certain shape that exactly fits with other types of proteins. So you can see here in the middle, antibody protein has a shape that exactly fits the protein from the flu virus, and so it can attach to that and recognize it and alert the body that it's there and to get rid of it. Alright, so the 4 levels of protein structure, and I'm sure you guys probably talked about this in the past, but let's go over it again. And yeah, so we have primary, secondary, tertiary, and quaternary structure. And so the primary structure of protein is a unique sequence of amino acids. So like each single monomer, so here we have the amino acid. Here's amino acids, so the unique structure, just the unique pattern of these. So, and it could include any of all these that are attached to this. So it's a unique sequence of amino acids. And then the secondary structure, which is found in most proteins, but not all, it depends on how big they are, consists of the coils and folds in the polypeptide chain. So when you put all the amino acids together, they might form like a helix, or here maybe there's. So here's 2 examples. So the most common ones are a helix or a pleated sheet. So it's kind of like a corrugated iron, or like something you'd see on the top of a shed, or like in a slum or something, like the roof, like the corrugated steel. So it's that's what like a pleated sheet looks like. And then the other option is a helix. So kind of what we talked about the carbohydrates before is when you join them together in a way, they might make some sort of pattern. So like when we talked about the alpha leakages in the carbohydrates, they made a helix when you connected them a certain way, but they were straight if you connected them with the beta connections. So depending on which amino acids you have, some of them may form a helix, so it'll curl up just because of the way they're connected, or they might form something like a pleated sheet just because of the way they're connected. And so then we'll move on to tertiary structure. So primary, sequence, secondary, the little shapes that they make with them together, and then tertiary is determined by the interactions among the various R groups. So here are all the R groups, like you remember. So like we talked about before, you have the nonpolar, polar, and like charged R groups. So like I said before, depending on how they're interacting with each other and how they are in the different structures, that's going to determine how the protein is going to take shape. So see here the tertiary structure. So you have the little pleated sheets and the helixes, but I guess you can see these little purple boxes within the pleated sheet and the helix. So those are going to interact with each other, either repel or attract each other. And so when you move on to the tertiary structure, they're going to fold a certain way, and that's going to make a certain shape. Either certain bands are going to be attracted, or they're going to be repulsed, or they're going to be attracted to water or repulsed by water, and that's how they're kind of going to fold together. So that's tertiary structure, how these secondary structures are interacting with each other to make a bigger structure. And then the quaternary structure results when a protein has multiple polypeptide chains. So you have multiple chains either coming together and interacting with each other, not just like the tertiary where these helixes and sheets and all those things are interacting with each other. You have like a whole nother strand. Like in the primary structure, you have a whole nother strand and a whole nother set of secondary structures and tertiary structures. You have those coming together to make an even bigger protein, and those interact, and that's the quaternary structure. So like two things coming together. Okay, so quaternary structure, two things coming together. Tertiary structure, the pleated sheets and helixes interacting. Secondary structure are these pleated sheets and helixes, depending on how you join the amino acid together, and the primary structure is each amino acid joined together. And you can also see in this picture, you can see like the different amino acids. You see the different amino acid R groups. So like alanine, if you start from the bottom, like alanine, protein, serine, glycine, arginine, valine, like all these different types, and you can see that up here in the R groups. Okay. So primary, secondary, tertiary, quaternary. Hey! Alright, so you can kind of see amino acid R groups. Again, what they look like. Alright, so the coils and folds of secondary structure result from the hydrogen bonds between repeating constituents of the polypeptide backbone. And the typical secondary structures in a coil are called alpha helixes and beta pleated sheets. We talked about this briefly. So here you go again. The abdominal bands of a spider secrete fibers made of a structural protein containing beta pleated sheets. Even though you can't see it there, you can guess it would be what? Beta, because that's what they are. Okay. And these, yeah, so there's a spider, and they're made up of proteins. Okay, the tertiary structure, it's going to go through this, is determined by interactions between R groups rather than interactions between backbone constituents. These interactions between the R groups include the hydrogen bonds, ionic bonds, hydrophobic interactions, like I said before. They might not want to be near the water, and then van der Waals interactions, which are just another chemical attraction, which is a little weaker than hydrogen or ionic bonds. And then strong covalent bonds are called disulfide bridges, and these reinforce the protein structure. Yeah, so it's just another type of a different type of attraction, so that helps stabilize between the groups that stabilize. Okay, now we're moving from tertiary to quaternary. So here's an example of, so like I said, you have whole new strands that are coming together, so it's the interactions between those that make an even bigger protein. So for example, collagen has 3 different strands So those all three different polypeptide chains, and those all interact. So the interactions between those three chains is the quaternary structure. And here's a hemoglobin that has two chains together, and the interaction between the light purple and the dark purple, that's the quaternary structure. So it results when two polypeptide chains form one macromolecule, and then like we said, collagen is a fibrous protein consisting of three polypeptides, and they're coiled like a rope. And then hemoglobin is a globular protein consisting of four polypeptides, two alpha and two beta chains. So there's actually two light purple components and two dark purple components there. Sorry. Okay, so just a little practical application. So you probably guys have heard of sickle cell anemia or sickle cell disease, and this is a result from a change in the primary structure of protein, which is what? The sequence of individual amino acids. Right. So a slight change in the primary structure can affect a protein structure and ability to function. So just changing one little amino acid can change the whole structure of the entire protein. So sickle cell disease, which is an inherited blood disorder, it results from a single amino acid substitution, so they're switched out in the protein hemoglobin, which we saw was the light purple and dark purple. Okay, so you can see number 6 here, glutamate is replaced by valine here. So just by replacing that, you can see how the purple and the red subunits change. And so when you integrate them into the quaternary structure, instead of having these nice round shapes, you have these more convoluted shapes in the sickle cell. And so as a result, when you have a normal hemoglobin, they're kind of round, so they kind of bounce off each other. But with a sickle cell, they're kind of convoluted, and they have like these hooks in them, or they're just they can like hook onto each other, so they kind of clump together. And so as a result, the whole red blood cells, they're inside red blood cells. This isn't the actual red blood cell, but you have all these hemoglobins inside the red blood cell, and that causes the red blood cell to kind of shrivel and form a sickle shape. So in healthy red blood cells, you have all these hemoglobins inside. They're kind of rolling around and bouncing around, and it's a normal round shape. But then in sickle cell, the hemoglobins clump together, and then the red blood shape kind of closes it on itself. Okay. And that was all just a change of what? One amino acid substitution. It can change the whole structure. Okay, so what determines protein structure? In addition to the primary structure, the physical and the chemical conditions can also affect the structure. So what's going on around the actual protein? So changes in pH, like we talked about previously, salt concentration, temperature, other environmental factors can cause a protein to unravel. So that's important because we talked about the protein will fold in on itself and things like that because of one reason is because the way it's responding to the environment. So if you change the environment, it'll change the way the protein is going to fold. So the loss of a protein's native structure, or the way it's supposed to fold, is called denaturation. And a denatured protein is biologically inactive, so it won't work. So here's a normal protein. You can denature it for a number of reasons. So like we said, pH, salt concentration, temperature, things like that. Denature, and then you have an unraveled denatured protein that's not going to be the same. However, you can restore those environmental factors and restore it back to the natural protein, which actually is pretty amazing because considering a protein can fold many different ways, but it just so happens to fold this way. But we'll talk about that a little bit. But yes, you can denature it take it away from the shape it's supposed to have, and then renature it, and it goes back to normal. Okay, so it's going to be hard to predict a protein structure from its primary structure, and most proteins probably go through several states on their way to a stable structure. And so chaperonins are protein molecules that assist the proper folding of other proteins. So here is a chaperonin, which is pretty cool, name at least. So it helps the protein fold. So here you have a polypeptide, just the sequence of amino acids, and it'll enter this hollow cylinder of chaperonin, has a little cap on it, it's going to come off. Put the polypeptide in, and then you put the cap back on, and then it changes the protein shape. The cap attaches, and then the cylinder changes shape in a way that it creates a hydrophilic environment for the folding of the polypeptide. So then in that way it can be like, oh, okay, so now I have to adjust. Now the polypeptide is to adjust so that the hydrophilic parts are on the outside and the hydrophobic parts on the inside. And so now the cap comes off and it just pops out and it's correctly folded. So that's what a chaperonin does. It helps it fold correctly. Okay, so just to kind of finish up, one way that scientists determine a protein structure is by using X-ray crystallography. It's just a method of seeing different proteins, what they look like, and so another method is using nuclear magnetic resonance spectroscopy that does not require protein crystallization. Bioinformatics uses computer programs to predict protein structure from amino acid sequences. So here's an experiment using an X-ray source. You put an X-ray beam through a crystallized protein. So basically you have to take a protein and crystallize it. And then that helps you to see it. Yeah. You guys don't need to know this. And so, but anyway, you don't need to know like exactly, you'll need to know this. But anyways, when you do this result, when you use X-ray crystallography, you're able to see different parts of the protein and different structures that you weren't previously able to, and because of this technology. Alright, so there you are for proteins.